Hi guys, in this video we're going to be talking about transformation. Now remember, a gene is a piece of DNA that codes for a protein or gives an organism a specific trait. So if we wanted to take a specific gene for something and put it in another organism, how would we do that? That's what transformation is all about. How do we get traits to show up that we want in different organisms? And there's a lot of neat applications for this, but first I'm going to go over some definitions. So genetic transformation literally means changes caused by genes, which is right here. And bacterial transformation, which is the type of transformation we'll be doing in class later on, is the process by which bacteria takes in DNA from its environment. And when it's incorporating that DNA, it can later express those genes that we want it to express. Now, this has got a lot of great applications in areas like agriculture. We can make certain plants or bacteria produce proteins that are like natural pesticides or pest resistant. We use this in the field of bioremediation. So we engineer bacteria that can uptake oil from the environment after an oil spill. It's really cool. And of course in medicine, there's a lot of neat applications, especially in gene therapy. All right, so we're going to start talking about bacterial transformation first. So, in bacterial transformation, what we're doing is we're putting a different piece of DNA into bacteria that it doesn't naturally already have. And if the DNA has a recognizable origin of replication, so a spot where it would normally be coding for that particular gene, and it's recognizable by the host DNA polymerase, then the bacteria will replicate the foreign DNA along with their own DNA. So it's like a little protein-making factory all on its own. It just needed those instructions, that piece of DNA. So we're going to usually do this with plasmids, and plasmids, if you haven't watched any other videos, are little circular pieces of DNA that generally are going to exist in smaller things like bacteria. So these are double-stranded, they're in a circle, and they're very small, they're not very many bases long, and hopefully, if it works, we'll incorporate the plasmid into the host, and then we'll have a transformed cell. Yay! And then when that particular bacterium starts to reproduce, it'll reproduce with that transformed plasmid as well. All right, so let's go through this step by step in a diagram. All right, so to get the gene that we want, first of all, we're going to have to isolate the DNA where it comes from. So I'm sure you guys heard us talk about GFP in class, green fluorescent protein. Now first we have to take that gene out of the jellyfish where it's normally occurring, that glowing gene, right? So we take that original DNA and we're going to cut it with restriction enzymes. Now remember, these are special enzymes that cut specific sequences in the DNA. So we'll cut our foreign DNA that has the gene that we want, and then we'll end up having this small little piece of DNA with sticky ends. And then we have our plasmid over here, our naturally occurring plasmid, that will also cut with restriction enzymes. And then that itself will have basically a spot for this new DNA to come in and have our sticky ends so that these will easily bind, and then we have this transformed plasmid. So DNA ligase, another special enzyme, is going to join our sticky ends together to form this new plasmid. Now we're going to put it into our bacteria. Hopefully it'll get transformed. And then we'll have our bacteria that's transformed with the recombinant plasmid. And then, like I said, hopefully, if all goes well, cloning will occur and the bacteria will reproduce with the recombinant plasmid and produce that gene and create those proteins that we want. And hopefully it'll glow. Yay! All right. So if we do this in the lab, and hopefully you guys will get a chance to do this, there's a couple of things we're going to do that may seem a little weird. One is we're going to incubate it in calcium chloride. And this is going to change the structure of our bacterial membrane, so pores will form when the temperature is raised to 42 degrees Celsius, and we call this a heat shock. Now, why do we want to do this? It's called making the bacteria more competent, making it able to take up that foreign DNA. And so hopefully that will work, and the heat shocking is a really fun process. And then, of course, we're going to be doing transformation with a selected marker. And generally, you can do this with ampicillin resistance or a glowing gene, so that if you were to plate the bacteria on a plate of ampicillin, you know, this is an antibiotic, it normally kills bacteria, but if it's been transformed with ampicillin resistance, that means it'll then grow, and you'll see these pretty little colonies. Also, hopefully, if it's taken up that transformed plasmid, that DNA, then we'll be able to see both glowing and growth on a plate with ampicillin. And generally, we're going to note this as plus if it has something, and then a minus if it doesn't. So on the plates where we don't put ampicillin, we'll put a minus, so no amp. All right, some key tips when you're doing this lab, you want to make sure you label the bottom of your plate. So if we have a Petri dish here, we take off the top, our plate that we'll label will be on the bottom, And of course, this is a much larger version than what you'll be using. You'll be wearing gloves and you won't be touching it all over the place. Touch it as little as possible. You're going to incubate it upside down after you've done all the procedures, and you're going to make sure it's nice and warm, so about 37 degrees Celsius, and you're going to do that for 16 to 24 hours. And then hopefully you can see the growth and see if you've had a transformation occur. All right, feel free to go back and rewatch if you need some more definitions. See you later.